shift gears here and talk about the ionic basis of a non-pacemaker cell. So this is going to be a typical contractile cell of muscle. This is not one that can autorhythmically or spontaneously depolarize. So for these cells, we have 5 phases of the action potential. Phase 0 is where we start with increased permeability to sodium. So we start out with an increased permeability to sodium, and then phase 1 is where we decrease that permeability. In phase 2, we're going to then increase permeability to calcium, and then we're going to decrease permeability to potassium. What this does, phase 2 is going to sort of stall the action potential. This is going to have movement of calcium, and we're going to block the movement of potassium here. This is going to allow for some time for calcium to enter the cytoplasm. And because with cardiac muscle, one of the features that we describe as a difference between cardiac muscle and skeletal muscle is that the source for calcium comes from the external extracellular fluid. It comes from the outside of the cell. So because of that, we need some time for that calcium to get into the cell. It's not like with skeletal muscle where the calcium is already in the SR, we just need to get it out. Here, we need it to get into the cell from the extracellular fluid. And so phase 2 is what we call the delay phase, which stunts or sort of stalls the action potential by preventing repolarization and allows time for calcium for the contraction to make its way into the cell. And then phase 3, we simply reverse the state of those 2 channels. We increase potassium's movement by opening up those channels. We decrease calcium's movement, and then now we can continue with repolarization. And then phase 4, we get back to resting membrane potential. Now, the duration of the contractile cell phase are about 250 to 300 milliseconds, so much, much longer than that of skeletal muscle for the reason that I just described, right? We need some time. We need much more time for enough calcium to move not only from the SR into the sarcoplasm, but also from the external environment as well, and for that to be able to have the events, which include the engagement with troponin and the bringing about of the myosin movement in order for muscle contraction, okay? So that is really important to have that delay, and this is why the contraction here is long. Okay, so let's illustrate those events here again. And I want to point out a very crucial difference between these cells. So they actually have a lower resting membrane potential. Their resting membrane potential is about -90. It's much more negative than it is for a typical neuron at -70, or even the pacemaker cell, which is also -70. So they are starting at -90 millivolts, which is way closer to potassium's equilibrium. And so we start out with movement of potassium. So there's movement of potassium and decreased movement of the other 2 ions, right? This is what's happening at rest. And from there, we start with phase 0, which is where we open up sodium channels. This is what's responsible for the rapid depolarization here. So the blue limb of this, phase 0, is the opening up of those sodium channels. Sodium rushes in, we depolarize the membrane. And then phase 1, we close those sodium channels to sort of begin stopping the, we're going to attempt to begin repolarization. But again, want to stall that for a bit. So phase 2, which is also called the plateau phase, we close those potassium channels that are already open to start. We close them here at phase 2. We open up calcium channels at phase 2, and this is the phase. Notice how this takes a bit of time. Okay, it takes a little bit more time, and that is allowing for the movement of calcium, which is crucial for the actual machinery of the cross-bridge cycling inside the muscle. So this window or this plateau phase is interrupting the repolarization in order to stall and allow for calcium to get in. From there, now we can continue that repolarization by closing those calcium channels, opening up potassium channels, and then further repolarize the cell back down to the resting membrane potential. So we want to just look at the big picture here. The major ionic movement that causes the upstroke of the action potential or the depolarization phase is sodium in this instance. It was calcium for the pacemaker cell, but it is sodium for the contractile cell. Very, very, very important. So the depolarization rather is caused by the movement of sodium. And this is similar to a typical neuron, right, in this instance. And then the other thing that's important to take away from this is the plateau phase. That's really important. The fact that we begin to depolarize by closing that sodium channel, but then we interrupt that. We close potassium channels because they were open all the time. And now we open up calcium channels, and then we can stall to allow for calcium to enter the cell, and then we can continue on with that repolarization. So here's just, again, a different way to describe this. We start at -90. Here we have potassium moving, nothing else moving. In this instance, then we open up sodium. Sodium causes the rapid upstroke or depolarization. Then we close sodium, but we still have potassium open, so we're going to start to repolarize. Then we interrupt that repolarization by closing potassium and opening calcium. That gives us the plateau or the sort of stalling phase. And then we close the calcium, close the calcium, open the potassium, and now we can continue on with that repolarization back down to -90 at rest. Okay. I'm going to pause here for just a bit and see what questions we have at this time. What questions do we have? Okay, very interesting question. Why are they called funny channels? I'm honestly not sure. But I think I read that the scientists that were studying this first noticed that they allowed movement of both ions and thought that they were somewhat atypical when we think about the traditional ion channel, which allows for either leaking of sodium or leaking of potassium. And so these unique channels actually allow for both those ions to move. So I think that that was the basis that I read, that because they were sort of atypical in allowing both ions to move, they were referred to as being funny. So they were kind of just a little bit misunderstood when they were first discovered. But if I find out anything more, I will let you guys know. But yeah, so funny channels allow both sodium and potassium, and they can depolarize because there's an abundance of sodium here. Okay. So let's think about the excitation contraction coupling and how that applies to cardiac muscle. So if you look at the properties of skeletal muscle, they're similar. So there's T-tubules in skeletal muscle. There are sarcoplasmic reticulum in storing for calcium. And then there's a troponin-tropomyosin regulation. So the binding of calcium, activation of tropomyosin, and that helps to regulate, turn the contraction on and off. So these are features that cardiac muscle shares with skeletal muscle, okay? On the other end, we want to think about features that cardiac muscle shares with smooth muscle. The gap junctions, that is one. That's a feature that we only see in smooth, especially single unit smooth muscle and cardiac muscle. And then the extracellular source of calcium for their contractions. So as we were just describing a second ago, the source for the calcium, that does this, that has this involvement with troponin, tropomyosin, and the regulation of the contraction, that calcium comes from two sources in smooth muscle and cardiac muscle. It's going to be the SR, but also the extracellular fluid. And so this is a shared property between smooth and cardiac muscle. So I typically, when I have time, I like to draw a little Venn diagram and look at the ways in which cardiac and skeletal muscle share properties, cardiac and smooth muscle share properties, skeletal and cardiac muscle. Like, look at the ways that each of those muscles share properties and then the ways that they're separate and individual and unique. But maybe try to do that as a study tool or try to organize that maybe through a table that can be helpful. But it can be interesting to see how each of these muscle types have things that they have in common with each other, and then things that they are very different or very unique with, right? Okay, so let's talk about the events of leading from the autorhythmic cell contraction to the contraction of a non-autorhythmic cell. So this is an autorhythmic cell. This would be a pacemaker cell. This would be a typical contractile cell. So just to reiterate here, the depolarization on this cell is due to the movement of calcium, the L-type or slow calcium channels. The depolarization on this cell is due to the movement of sodium, right? So two different types of cells, and there's two different ionic movements that result in their action potentials. And so we start with the contraction on this cell, or the impulse on this cell. We spread that electrical impulse through the gap junctions, and that is how it gets to a contractile cell. So it's really important to remind ourselves that these cells are not autorhythmic. They need to receive the impulse from an autorhythmic cell. So once that impulse comes in, then we can begin to see calcium moving into the cell from two sources. One is from the extracellular fluid, and the other is from the SR. So both of these sources will help to bring calcium into the cell. This is why it's so important for the plateau phase in this cell's contraction. That plateau phase allows enough time for calcium to get from the extracellular fluid, more importantly, but also from the SR into the sarcoplasm. From there, it can bind to troponin, and then we have the same regulatory events that moves the tropomyosin away and uncovers the binding sites. Myosin can bind to those sites on actin, and then we can bring about cross-bridge cycling. And then when it's time for this muscle to relax, we're going to have two sources of ridding the sarcoplasm of calcium as well. Because we had two sources to bring calcium in, we also have two different ways to help calcium leave the cell as well. Actually, three ways, because we have two ways that it can leave the cell entirely, and then it can also go back into the SR. So one is via the calcium ATPase pumps that pumps it back into the SR. Two is via the calcium ATPase pumps that pumps it out of the cell. And then three is by the sodium calcium exchanger. So this is an example of a secondary active transport, whereas this is primary active transport. If you could think way back to module 1 or 2, I think we did talk about the different types of transport, right? Active transport can be primary if it's relying on ATP, or secondary if it's relying on the movement of another ion. So both of these are examples of ways that we can get the calcium out of the sarcoplasm and then help that muscle to relax. Okay, and then we can go back into the relaxed state and cease the contraction. So just reiterating that here, relaxation of cardiac muscle happens by three mechanisms: the calcium ATPase pump on the sarcoplasmic reticulum, the calcium ATPase pump on the plasma membrane of the muscle to send it out of the cell, and then the sodium calcium exchanger that is also on the plasma membrane. And so this helps to return troponin back to its state where it's covering up the binding sites, and then that helps to disengage cross-bridge cycling. Okay. Now, another little concept we want to discuss here is we want to think about the electrocardiogram. So this is a way that we can actually measure and record the electrical activity of the heart. This is recorded by electrodes that we place on the skin. So if you go to your physician's office and they send you for electrocardiogram, the technician is going to start by placing leads on your arms, on your chest, on your legs. And if any of you have had this, you know exactly what this procedure looks like. And it's non-invasive, so these leads are not sort of piercing the skin, they're sort of on your skin. And this can be a way to detect any abnormalities with your electrical rate and rhythm. So if you have a pacemaker defect, if you have what we call a heart block or one of the rhythm defects, you will have a delayed conduction, or you may have a slowed heart rate, or sometimes even a fast heart rate, depending on the type of defect, right? If you have an atrial fibrillation, for example, your atria are just spasming, right? They're not contracting synchronously, and that is an example of one of those rhythmic defects. So that will be detected by way of the electrocardiogram. So it's an external measure of the electrical activity that's happening in the heart. The body is used as a conductor, so it's leads that are placed on the skin. And we can do this for other organs as well. So the ECG is for the heart, but we can also look at the EMG, the electromyogram for muscle, or the electroencephalogram for the brain. So this procedure is not unique to the heart, but it can be used in other parts of the body to detect conduction of impulses as well. Now, the distance or the amplitude of the spread will be based upon the size of the action potentials and the synchronicity, how well they're synchronizing the events with other cells. So this is what the test is really looking for. How big those impulses are the impulses large enough? Are the action potentials the correct magnitude? Are these events being synchronized with the appropriate chambers? And that is what we would detect on the test. And so this is going to make sure that the heart electrical activity is synchronized, the atria contract fully, then the ventricles. And if we don't have that, we can have many different types of defect. We can have a bundle block. We can have several defects that would throw that synchronized rhythm. Now, quickly, we want to think about the phases of that electrical activity. So we have the P wave, which is the atrial depolarization. Remember, that has to happen first. QRX is the ventricular depolarization, and that is overlapping with the relaxation of the atria, which is the atrial depolarization. The T wave is the ventricular depolarization. So now the ventricles relax. And then the PQ segment is the AV nodal delay, the space in between the time it takes for the atria to contract and the ventricles to contract. And then we can think about the QT interval between Q and T as the full ventricular ejection, and the TQ segment, which is between T coming around to the next Q, right, of the next segment, we call that the TQ. That would be an example of the ventricular diastole, or the ventricular relaxation. So this will make more sense when we talk about the cardiac cycle, which is our next topic that we're going to discuss coming up, all right, on Friday.